The following content is provided by the students of King Saud bin Abdulaziz University for Health Sciences, as a part of the College of Applied Medical Sciences Virtual Academy. Your support will help us to continue providing educational contents. The basis of genetics is formed by a macromolecule of carbons, nitrogens, oxygens, and phosphorus. This macromolecule is called DNA. Or as Crick and Watson referred to it, the secret of life. The story of genetics started off in 1865 when Gregor Mendel proposed a pattern of inheritance and established Mendel's laws of inheritance. Since then a lot of things have happened, such as the theory of the transforming principle by Frederick Griffith in 1928. Then, the discovery of the molecular structure of the DNA by Crick and Watson in 1953. After 17 years, Francis Crick published his paper about the central dogma of molecular biology. In 1990, a group of scientists started working on human genome sequencing. They finished sequencing the entire human genome in 2003. The previously mentioned discoveries opened the door in front of researchers, which in turn resulted in the innovation of many clinical applications in the field of genetics. Many of these applications involve the use of DNA. However, DNA can be found only inside the nucleus, therefore, a procedure to extract the DNA is required. There are many methods to extract DNA, but the most widely used ones are organic isolation method, inorganic isolation method, solid phase isolation method. We use an organic method because it works for us, and for its safety. This video will focus on extracting the DNA from whole blood samples. In whole blood samples, the DNA is found inside nucleus of white blood cells because it is the only nucleated cellular component. This is the list of materials you need. Add 900 microliter of your cell lysis solution. Then add 300 microliter of the blood to the same tube. Notice the white pellet at the bottom of the tube. If it is there, discard the supernatant, leaving only 10 to 20 microliter of residual liquid with the white pellet. Then, resuspend the white pellet in the residual liquid. After completely resuspending the pellet, add 300 microliter of nucleolysis solution. Mix the solution with the pipette and notice the apparent viscosity. Now, add 100 microliter of protein precipitation solution to the viscous solution. Vortex. Notice the brown pellet. If the brown pellet is apparent, then transfer the supernatant to a new tube. Then add 300 microliter of isopropanol to the supernatant. Notice the white pellet. If it is there, discard the supernatant. Then add 300 microliter of 70% ethanol. Discard the supernatant but with care because the pellet is loose now. Now invert the tube and let it air dry for 10 to 15 minutes. After completely drying your pellet, add 100 microliter of DNA rehydration solution.